Hi, my name is Niloufar Namovari, and I'm going to present my poster titled Investigation of a TRIM28 gene variant identified in a pediatric patient presenting with intellectual disability. TRIM28 is an epigenetic repressor protein that is recruited to DNA by CRAB-ZFP transcription factors and then recruits other repressor proteins, including ATRX. This repressor complex as a whole suppresses these targeted regions by adding or removing epigenetic marks. TRIM28 targets transposable elements, a class of repetitive elements that can jump from one region to another, but usually their activation is suppressed. Activation of transposable elements can cause mutation, triggers an immune response, or dysregulate the expression of nearby genes. Activation of transposable elements has been linked to brain disorders, and previous studies have found that TRIM28 depletion in human neural progenitor cells derived from induced pluripotent stem cells activates transposable elements and alters the expression of neural genes. A few years ago, a local male pediatric patient presenting with intellectual disability, behavioral issues, febrile seizures, dysmorphic facial features has been brought to pediatric clinic at Victoria Hospital. Based on his clinical features, he was initially suspected to have ATRX syndrome. But when they did exome sequencing, they found that it's not ATRX syndrome. It's actually 2123 T2C variant in exon 15 of TRIM28 gene. Given that ATRX and TRIM28 are both in the same complex suppressing transposable elements and based on similarity at the clinical level, we hypothesized that 2123 T2C TRIM28 variant might have similar deleterious outcome as ATRX mutation in brain development due to reactivation of transposable elements. To begin with, healthy donors' somatic cells has been reprogrammed to healthy donors' derived iPS cells. And this specific variant in TRIM28 gene has been introduced to healthy donors' derived iPS cell using CRISPR-Cas9 technology. So I will be using this TRIM28 mutant iPS cell line as my disease model and healthy donors' derived iPS cell line as control. First, I was interested in studying the normal pattern of TRIM28 expression at RNA and protein level during neural induction. So I use a protocol based on dual SMAD inhibition to induce iPSC control cell line toward neuroepithelial stem cells. First, I confirmed pluripotency of this iPSC control cell line using immunofluorescence staining. And then every 3 days during neural induction, I looked at expression of TRIM28 at RNA level and protein level using real-time qPCR and Western blot analysis. Here you can see that this iPS cell line is actually pluripotent. We can see expression of NANOG and SOX2 in almost all cells of this colony, these iPSC colonies. And here is the result of qPCR. First, you can see that NANOG is decreasing during neural induction, which means these cells are not pluripotent anymore and they are actually differentiating. And we can see an overall decreased pattern of TRIM28 expression at RNA level. Here is the result of Western blot analysis. And we can see an overall increased pattern of TRIM28 expression at protein level. This opposite pattern in TRIM28 expression at protein level and RNA level could suggest a post-transcriptional regulation of TRIM28 during neural induction. The result of this study will be confirmed using more replicates. I'm also interested in comparing transcriptome and chromatin accessibility of TRIM28 mutant neural progenitor cells derived from iPSCs with control neural progenitor cells derived from iPSCs. Because in silico analysis of this specific variant has shown that, has predicted that probably this variant is detrimental to protein structure and function, we expect to see activation of transposable elements in mutant neuroprogenitor cells, and also more openness toward transposable elements in this cell line in comparison to control.